This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
All right. So everybody, this is the Unsupervised Learning podcast, and we have a returning guest, a many-time returning guest. And, I can see the traffic, and I know that this is a popular guest. It's Dr. John Hawks, University of Wisconsin, paleoanthropologist, co-author of a book with Lee Berger, and I know you're working on a textbook right now, commentator on things naledi, Flores, archaic integration, many, many different topics. I've known John from the internet for over 20 years now, I think about 20 years, and, we have met several times in person, and so, John, I think you can say we're friends, and we have shared similar interests actually for a while now. And some of your writings have really influenced, I think, my thought that percolates into my Substack and my commentary that people have listened to. So, we're going to explore some particular topic today, actually, in some depth, about archaic admixture and what we know in 2025, which is so much more than what we knew in 2005, when I feel that, to be honest and candid, and I'm not saying this is your friend, you were ahead of the curve. You and Greg Cochran in some of your works together as well, Greg Cochran as well, but with your mentor Milford Wolpoff, the multi-regional hypothesis, those sorts of ideas, that model, you were ahead of the curve in anticipating the possibility of archaic integration, adaptation, admixture, what it means to be human species, and all these things. And I'm throwing a lot out there. Can you take some of the, because some of the listeners are probably 30, and so they were 5.
That's right. Yeah, no, it's crazy. It is totally crazy. Well, listen, thanks for having me back. This is a topic.
Take us back. Yeah.
I've been thinking about this topic, as you said, for a very long time. I'm really, really engaged in it right now. A couple reasons, right? One is that my book I'm working on now is a modern human origins book. And we're really, I'm focusing in on those factors that caused the people that you see around the world today to be a great success, why African groups were able to, sort of put something together that hadn't been put together before, and what happened when they encountered these archaic human groups, initially within Africa, but then as they emerged into Eurasia, they encountered the Neanderthals, the groups that we call Denisovans, and there was this genetic mixture that happened. So understanding, the scenario in which that happened, and the reasons why there were, potentially, synergistic combinations of DNA that emerged, that's really what I've been focused on. So, you invite me to go way back. I'll tell you, when I was a student during the 1990s, the framework of how we looked at what's going on with today's people around the world, what our more immediate origin and ancestry had been, was focused around this very strong debate between the idea that we came from Africa, all of us, in some sort of relatively geologically recent event. And today we understand that that's an event that's the last 100,000 years of time. Versus we had ancestry from many different populations from different parts of the world, populations that existed in Africa, that existed in Eurasia at the time, including the Neanderthals, including archaic peoples from East Asia, from Southeast Asia. That was the multi-regional idea. And as the evidence developed through the late 90s into the early 2000s, it became pretty clear that the out of Africa idea was substantially correct, that most of our genetic heritage had come from Africa in a recent time, but that didn't seem to explain everything. there were still these features of skulls that were, that seemed like they showed some continuity over time in Europe, in East Asia, in Southeast Asia, Australia. And there were little pieces of the genetic record that seemed to point to a surviving diversity of earlier people. And so, in the early 2000s, when I was working on this, those were the things that were occupying me. We were trying to account for this pattern of genetic diversity that had emerged in the course of the Human Genome Project. As the Human Genome Project completed our genome, the first step after that was to add diversity to our knowledge of genetics. And that began with genetic samples that represented European, East Asian, and African populations, but it expanded after that to a broader array of populations. And as we were getting those data, you would find genes that seemed like they had a deeper diversity than would have been expected under the out of Africa scenario. And so my idea at that time was that, well, maybe we're getting some genetics from Neanderthals or from other ancient groups that we're picking up in these genomes of today's people. What would that look like, and how would it work? And so the idea of introgression, which was a long-standing idea in biology and in genetics, especially coming from botany, it was something that human geneticists hadn't thought about a lot. I can tell you that our standpoint from the multi-regional model had been that we're looking at something like isolation by distance in populations of humans over hundreds of thousands of years. And that didn't lead us to think that we were going to look at some sort of, extraordinary shift in gene frequencies that had happened in the recent past. The genome data by the early 2000s told us there was this huge shift of allele frequencies that had happened in recent time, but there were still patches of more ancient diversity. And modeling what that should look like, that's when we brought in the introgression model. And so that was, where I was thinking 20 years ago.
Yeah, and I will say, Neanderthal genome shows up. Neanderthal genome shows up, and we see that there's admixture and all this in 2010, and everyone's like, "Oh my god," blah blah. And we've talked about this before, but I want to say this. Before that happened, people would say you're crazy for thinking that we would have sex with Neanderthals, that there was admixture, and then it was one of those things. After it happened, people started saying, "Well, we always believed that." I was there. You were
in
there.
my career.
Yeah,
I love that you say that, because I've been through so many things in my career where I've seen the stages and the initial these stages of acceptance of a scientific theory, right? At first, it can't possibly be true. And then, well, maybe it makes some sense, but the data still aren't there. And then it's, oh, yeah, that makes sense, and we knew it all along, right? and we definitely went through that with the ancient genome data made some things really, really clear really quickly. Those early genomes, the Vindia genomes that were, sort of a combination of 3 genomes. So it was published in 2010.
That's a Croatian, just a Croatian Neanderthals. Those are Neanderthals from Croatia, just so you know.
Yes, Neanderthals from Croatia. And then immediately after that, the Denisovan 3 genome, right, which was this other population, this Denisovan population, as it became called, they made a couple of things clear immediately. One is that people living today have some part of these genomes, and the some part was really small. It was something like, at the time, estimated 1 to 4% from Neanderthals. And today we aim at a number that's something like a little under 2%, varying a little bit from group to group in Eurasia and the rest of the world, a lot less than that in Africa. Something like, 1/3 to 2/3% in different parts of Africa. So that became clear really quickly. The presence of this Denisova-like population became clear immediately because Svante Pabo's group, who was sequencing the DNA of the archaic genomes, had the foresight to include a sample from Papua New Guinea in their initial sample of human genomes. I think something looking back that it's maybe tough to imagine today, especially if you're familiar with genetics, is that in 2010, we had very few genomes of modern people to work with.
There was an Excel spreadsheet. There was an Excel spreadsheet listing every single individual.
Yeah, absolutely.
It was dozens.
And almost all of those individuals were European individuals. And so when Svante published these Neanderthal genomes, it was a major contribution to publish. I think they had in their initial round 5 genomes from less represented peoples from different parts of the world. And one of those was from Papua New Guinea. And so finding that there was this genetic similarity between this Denisova genome, this site in Siberia, and somebody in Papua New Guinea was a surprise. It was not something that people expected, but it clarified right away that, this genetic heritage is in places that are not necessarily real close to where the ancient genomes are being found. And that was also true of the Neanderthals, right? The 1% to 4%. Anthropologists like me at the time were surprised that this was just as high, and in fact, we understand today a little bit higher in East Asia than it was in Europe. The expectation, I think, that most people had was that this was going to be regionally focused in the parts of the world where Neanderthals lived, which was Western Eurasia. And it is not that. It is globally present in all of the populations that are outside of Africa.
Yeah, so I think, you're talking about some of our expectations, and what I will say is, I think you had a good theoretical model that anticipated what we see today. But devil's in the details. There's a lot of interesting things in the details that you didn't anticipate, that I didn't anticipate, and we couldn't anticipate. We wouldn't, we had a very, very simple model, like the physicist with the spherical cow. It's we admix with we know now that, for example, even though the overwhelming majority of Neanderthal admixture in modern humans is from a single event, it seems, probably in the highlands of West Asia, there were other admixture events. It just happens to be, for example, this is confusing to a lot of people, but in many parts of the world, the first settlers left no imprint. And so if you have later people coming from the Near East, as occurred in Europe, you don't have the imprint of the European Neanderthals, which the initial upper Paleolithic people, modern humans that are coming out of Africa, actually did, right?
That's exactly right. I think, this is such an important point. The entire world was full of small-scale, very sparse populations, right? They're hunter-gatherer societies. They're living in groups that are probably foraging bands that are 20, 25, maybe 30, 35 individuals. And every one of those is tenuous. And that was true of the Neanderthals. It was true of the Denisovans. It was true of modern people. It was true of everybody in Africa, right? Everybody was tenuous. And the bones that we find as anthropologists are always bones from somebody that likely didn't make it in the long run. And so I like to use the example of people of the Vikings in North America, right? We understand today, and we had historic records, that there was this Viking settlement in North America, and it fundamentally left almost no archaeological trace. Extraordinarily difficult for archaeologists in the end to track down. And if it hadn't been for the historic record of this in the sagas, we wouldn't have known about it probably from genetics or any other source of information, right? Most contacts between populations were like that. They were real contacts. They probably involved intermarriage, almost all of those contacts in the past. They involved groups that lived with each other, knew each other, got to know each other, right? We're not talking about, when the African-derived populations are finally dispersing throughout the rest of the world sometime after 50,000 years ago, we're not talking about, strangers showing up that you never heard about before because they were thousands of miles away when your parents lived. We're talking about people that were always in contact. And so every time those groups are in contact, children are born. And we find the evidence of groups that incorporated those children. And those children lived for a while, and that group became successful initially, and then in the end failed. And that happened again and again and again and again. With almost all of these groups that existed in these periods of time are groups that did not become ancestors of ours. And tracing back what a geneticist would call the coalescent through the ancestors of ours, we're never finding those people in the archaeological record. We're finding the cousins of those people. And so, we're trying to trace back what happened to our ancestors, knowing that they're part of a world with a lot of people that we're finding that did not succeed.
Yeah, I mean, I think one way people could think about it is, you have these like genealogies in the past that did exist, and we're sampling in those genealogies to kind of create a scaffold of the trajectory of human demographic population history. Just take it seriously, not literally, is one thing that I would say. I want to bring up in terms of admixture and archaic introgression and all these things. One of the things that I'm sure you've been keeping track of, I mean, I know you have, and I have, is the fascinating reality that it looks like Neanderthals themselves received early admixture from a population that was related, that split off from the stem modern human lineage, the out of Africa population. And here's for the listener or viewer, for the listener, one of the methods that I thought was pretty ingenious. There's multiple ones. One of them that I thought was pretty ingenious was they actually looked for Neanderthal-like segments in the genomes of people of unadmixed African heritage, and they found that those Neanderthal-like segments were not actually Neanderthal-like segments.
Mm-hmm.
They were like segments that Neanderthals had in them that were from a distant, unsampled group that probably left Africa like 300,000 years ago, right? So can you talk a little bit about that?
I would love to, because one of the things that I think people, if you're watching this field, even if you're watching it sort of closely, it may be impenetrable how the evidence base has shifted over time as a result of analytical changes. Our data have gotten better, right? We have high coverage genomes from, I think now 4 or 5 Neanderthals, from still just the one Denisovan. But we have low coverage genomes from a larger number in both cases. And so we know more about their variation than we did before. And of course, we have a lot of genetic information now from modern people who were after the Neanderthals, after the Denisovans, but in some cases very early afterwards. And so that's helped us to understand, okay, what does it look like when we have an ancient, haplotype that hasn't been in a human population real long? We have some genomes that show us that now. But a big shift has been in the initial phases of ancient DNA using short read technologies, which are what we have to use with ancient DNA because it's all broken up into little short pieces. It was very difficult to understand what longer haplotypes would look like. And so some of the big tools for us to understand the genealogical connections between people are haplotype-based tools. We need to know how long the segments are that they share. And it became really tough to do that with Neanderthal genomes until human genomes got sampled in enough numbers that you could start to reconstruct these, phase them, as it's called, into haplotypes from short read data. And once that became possible, people were able to apply more and more sort of sophisticated statistics to find out when Neanderthal haplotypes came into populations, how long are the haplotypes that we share with them, and are there mismatches, right? Are people sharing things that are sort of the wrong length? Because there's a characteristic length of haplotypes that they ought to be if they're coming into our population 50,000 years ago or going into their population at that time. And that's what you're talking about, right? As people began to get more and more samples of today's people, they were like, well, actually, we knew from the early stages, right? Using the F4 statistics that David Reich's group really pioneered, Nick Patterson, David, other collaborators of theirs.
Give a quick sketch with the F4 population statistics.
Yeah, exactly. So if you know nothing at all about the haplotype structure of a genome, one thing that you can do is just count how many times two people mismatch each other. And you can create a hierarchy of your samples so that I'm comparing two people with one ancient genome. And then I'm going to compare them also with an outgroup. And that makes 4 genomes that you're comparing. And that gives rise to what's called an F4 statistic. And the whole principle of this is I don't really need to know where these genes came from. I just need to know if one of these people is more like the archaic genome than the other. And if that's consistently the case, then that tells me that this one person has some sort of genetic connection with that archaic genome. So that was a really simple kind of approach. It was a vast improvement over what we had before that, which really depended on a lot of population genetic knowledge to apply. This required no population genetic knowledge, and it turned out to be a really great statistic that allowed us to study admixture in lots of situations. But one thing that it didn't do especially well is to give us an idea of how long ago things happened. And so and to their credit, right, sampling lots of archaic genomes gives you and lots of modern genomes over time gives you a way to structure your sample so that you can talk about time. So you can figure out when things happened by sampling the right time. But with deep time, that becomes harder and harder to do. So so that's the F4 statistic. As we got working on these Neanderthals with that statistic, it became clear that Neanderthals and Africans were too close to each other compared to Denisovans, right? We knew that Neanderthals and Denisovans were sisters. And if they were really sisters, if they really shared a common ancestry, and they branched off from the African branch from a common ancestor, then Neanderthals and Denisovans should be equally close to us. They shouldn't one of them shouldn't be closer than the other. And actually, the Neanderthals are closer to Africans than the Denisovans were, and that was a mismatch. It became clear when haplotype sort of structured methods became more and more available, became clear that was happening because Neanderthals actually got a bunch of stuff from Africa. And so that was, we know a good bit about when this happened because we've got time samples of Neanderthals that help us to figure out when it happened. The Cima de los Huesos sample of an early Neanderthal population, they existed about 430,000 years ago. They don't have this African-like mixture. Neanderthals that lived within the last 200,000 years did. And so we know that there was a shift, that sometime before 200,000 years ago, some African-like genetic mixture happened, moving African-like haplotypes into Neanderthals, replacing their mitochondrial genome and their Y chromosome wholesale, and bringing with it something like 10% of the Neanderthals genome.
Yeah, the Y and the mtDNA thing is weird. I mean, honestly, David Reich, ancient DNA, Svengali, Empresario, I don't know what kind of word you want to use. But, he straight up has said, and I've gotten this sense from people in his lab where they feel like there's something broken in our current model because this replacement, doesn't have like a great explanation, like why it would selectively replace. Like there could be selection on the Y and the mtDNA, maybe. There's ways that you can imagine it happening, like in sequence, but it seems pretty weird. Like, do you have any thoughts on that?
I was writing about selection on mitochondrial DNA in the 90s. And so that doesn't bother me. I think the mitochondrial DNA and the Y chromosome both, what they share, they have different mutational rates, right? The mitochondrial DNA has a faster mutation rate than anything in the nuclear genome. But the Y chromosome is a lot longer, and it's similarly, tightly linked for the most part. So if you inherit a bad allele anywhere on your Y or anywhere on your mitochondrial DNA, that's bad, right? And there's no chance for recombination to solve that problem by bringing in something good to combine with it. So there's this constant attrition of mitochondrial lineages, Y chromosome lineages due to purifying selection. And there's also the constant possibility that a population has run down enough, right? This sort of mutational ratchet has gone to the point that any change that makes a positive improvement would be rapidly selected. So you've got purifying selection that's pretty hard, and across them, right, enough of a mutation rate that a ratchet might actually make things worse over time. But any rescue mutation that would happen then has a big advantage. So that leads to, I think, a faster cycling of selection on both of those systems. So I'm relatively happy with that. But the mitochondrial timeline in particular has had, sort of this interesting history for us from the fossil standpoint, because it was initially the thing that we had to figure out when did Neanderthals diverge from African groups, right? And so there was a lot of thinking about Neanderthals that happened under the constraint, under the expectation that Neanderthals and modern humans had diversified something like 300,000 years or less from each other, right? The big story about mitochondrial Eve was a story that tied into that timeline. And we know today that timeline is artificially short, that the mitochondrial sort of ancestor of the earliest Neanderthals and the Denisovans and us was something like 700,000 to 1 million years. And so you have this sort of longer timeline available for the fossil record now. And we're still trying to align the fossil record with it. That alignment between the genetic timeline and the fossil timeline is still uncertain at that earliest sort of part of Neanderthal Denisovan evolution. Really interesting questions like the famous fossils from Zhoukoutian, China, which were called Peking man historically. They're assigned to Homo erectus. They're in a time frame, something like 550 to 750,000 years ago, that probably is too early to be Denisovan, but maybe not, depending on how that timeline actually is aligned. And so those sorts of questions of what was the early history of these populations? Can we connect them to the fossil record? Maybe we'll get ancient DNA from some of these or ancient protein data that will help to triangulate a little. But until then, we're going to have some challenges.
Yeah. There's so much there. Can I ask you? You're a fossils guy. You know the fossils. How shocked were you about the revelation of the Denisovans in 2010? I asked partly because I was reading this stuff in the 1990s as well, and there was always something fishy about East Asia. Here be hominins, or hominids as we called them then. It was just like people knew there was stuff there, but they didn't know what. And I'm just like, okay, I was pretty surprised or interested, but I was also partly well, I mean, yeah, it kind of makes sense. There was something there.
East Asia, Southeast Asia, and Africa, all three of those regions suffer from the same problem relative to the Neanderthals. With the Neanderthals, there was a very tight, strong record of them going up into radiocarbon times. So you knew what was going on with the last Neanderthals pretty well chronologically. And even before radiocarbon times, so sort of times before at that time, 40,000 years ago, even when you got back into that sort of pre-radiocarbon era, you had a glacial series that in Europe, people felt like they understood pretty well. And so you could sort of, the linking of the hominin fossils, the faunal bio sort of chronology of different species in Europe, the technological series, right? It became possible to develop a timeline that was pretty good within the last 120,000 years at least. That did not exist in East Asia, in Southeast Asia, in Africa in the same way. The interesting things in all three regions were all well before radiocarbon. And the density of the record that was available, especially for European and American archaeologists to read, was just not enough to make sense of a chronology. in the same way, right? We knew that things were Middle Pleistocene or Late Pleistocene. But to talk about what was going on in a finely resolved sense, you couldn't do it. And that was a challenge for the early discussion of the out of Africa model, because it was hard to pick out which African fossils were actually relevant to this. People are today, we understand that the oldest of these sort of early pre-modern hominin populations in Africa is from Morocco, from Jebel or Hood. But in the 90s and early 2000s, those fossils were thought to be late in this game. They were 35 to 50,000 years old. They were too late to factor in. And so you sort of had this problem of chronology. In East and Southeast Asia, what have turned out to be the central fossil samples in this conversation are fossil samples in Southeast Asia from Guangdong. And we knew that was the latest of the erectus-looking fossil samples in Southeast Asia. But we did not know at that time that they were 100,000 years old. So that was, putting them into this time frame of, maybe these are the people that actually were there when African descendants showed up. Those are likely to, in today's framework, to be some of these Denisovans. Likewise, in East Asia, the main fossil samples we know today actually perfectly well, right? The Harbin skull that was identified in 2021 as something that was, that first revealed to science, looks a lot like many other fossils from East Asia from probably the same time frame. It's something like 160 to 200,000 years old. And it's got, we know today, a Denisovan mitochondrial sequence. And so that tells us, okay, this group that we call Denisovans is characterized by this kind of anatomy in China. And this group that we call Denisovans has deep divergences. We know about those genetically. And those deep divergences might incorporate some of these Southeast Asian fossil groups, right? So today, it makes a lot of sense. When the Denisovan genome was first revealed in 2010, it came out of left field because it was in Siberia. and so it became this question of, there is something very different from Neanderthals. And that something has relatives that contributed to populations of extreme Southeast Asia. And as it has become clear, across much of mainland East Asia as well, at a very, very small percentage. And so there's every reason to think in that era that these are going to be the fossils that we have already found in China and Southeast Asia. But tying those together was something that wasn't really easy.
So, we were talking about Southeast Asia. One of the things that fascinates me is how speciose Southeast Asia was, and, it doesn't seem like there's been any, never say never, been any integration. There could be super archaic admixture. You can talk about what that is. But, we have all these different human species in Southeast Asia. What's up with that?
Well, I wish I could say it was as simple as islands. The islands are a big part of it. And this is a part of the world where you have repeated, as every time sea level is high as it is today, you have islands. And every time the glaciers advance across the northern hemisphere, the sea level falls and many of those islands become connected to each other. And so you have this biogeographic situation where you have repeated isolation over, 100,000 year time frame. And so that's one element, right? You have an isolating mechanism that in evolutionary terms actually might create species. Then you have the fact that some of those populations got onto a boat sometimes and went to an island that was never connected to the mainland, and they went their own way. And that's what we see with the Philippines, with the population that we call Homo luzonensis today, with Flores, the ones we call Homo floresensis. Islands seem like that's the explanation of these. But we also have, the challenge that the samples that we have are really limited, right? So in Java, for instance, right, we got a lot of fossil hominids from Java. It has been, a really good environment because of the geological uplift and the creation of these sort of sediment bowls across Java to catch some of these hominids and to sample them across time. And so we can see the evolution there to some extent. It's not clear whether the end of that sequence, the hominins from Nandong in Java, which are 106 to 120,000 years old, they've got very erectus-looking skulls, except they're bigger than any of the other erectus skulls in terms of brain size. It's not clear to us whether this is the end of an island lineage that has sort of evolved across time in this area, or whether you've got significant genetic input from other populations that might be more connected with East Asia, with Europe, with Africa, or whether this is actually just Denisovans and they've picked up some local thing when they got to island Southeast Asia that gives them this sort of skull look that maybe they didn't have in other places that they lived. I wish I knew. It's one of the real challenges of the fossil record. I was writing about this today, that we, from the beginning, assumed that the skulls could tell us something about how populations were connected, right? Studying similarities between skulls tells us something about their genetic similarities, but one gene might make a lot of similarity happen, or one gene might make a lot of difference happen. In other words, how similar your skull is not a very good measure of your genetic similarity, right? It might tell you you've got something that you share, but it doesn't tell you necessarily how much you share.
Yeah. I mean, we're still, I guess it's been long enough. Like, is your hope for ancient DNA from the hobbits? Are you over it? What do you think? I mean, you're never 100%, but I'm just saying.
I think we're going to have ancient DNA from the hobbits. I really do. Right? They lived less than 100,000 years ago.
Okay.
I think it's gonna come into scope. Right now, the challenge is not that there's not DNA there. The challenge is that the DNA is in segments that are so short that we can't reliably tell whether they belong there or not. So it's in large part not it's not the case that there's no biochemical preservation. It is the case that the biochemical preservation doesn't accord with our ability to verify it. Protein data, I think that we're very likely to have. I think we're going to have protein data from all these. And the protein data is very limited, right? For folks that are listening, there's not endless hope of great sort of phylogenetic resolution from protein data, right? We're going to find that they share one or two proteins or one or two amino acid changes, across their whole, the enamel proteome of some of these species is like 300 amino acids. So it's not a lot of information to work with. We're dealing with that in South Africa in the work that we're doing. This really, really cool protein information that we get.
We've gotten greedy. Like the things that you're saying, you know what I'm saying? Like 30 years ago,
yeah,
you would just be like, wait, what am I saying here? 300 proteins, that's not a lot of information. That's 300 variables. You would just be like, what?
Yeah, I know, right? If you compare that to the very first mitochondrial sequences, which were just the hypervariable region 1. So that's a 300 base pair segment of DNA that was the first ancient DNA information that people worked with through, the years from, let's say, 1997 to 2002, 2003. That was the main thing. And it's a 300 base pair piece of information. So having 300 amino acids is not bad. It's pretty good. But I'm actually I just feel like we're just getting better with excavation methods. We're getting better at recognizing where DNA is preserved in samples, and it's getting cheaper. And if there's one thing that's going to solve some of these problems, it's being able to throw biochemistry at thousands and thousands of samples as opposed to, 2 samples, right? Yes, if we have to drill that tooth, we're not going to get very much information from it. But if we can take every tiny piece of bone that is in the sediment from a site and be sure that we're getting whatever's in it, we're going to get it.
Well, I mean, do you think we are at, look, it's like diminishing returns. Like, are we at the number of like branches of the human family tree? And, you might want to talk about some of the arguments about species names and all this stuff. I lean more towards your side. I think more geneticists lean towards your side, where you tend to have an expansive view of species. I got into this like when Dragon Man first came out. I'm just like, what are we thinking? Like, it's clearly Denisovans. And I got like, people were like, you can't say that. How do you know? And I'm just like, what else could it be? Like, if it like, it's everything, it's circumstantial evidence. I was right? I was right in terms of like, I'm not saying that it was a deep insight. I think a lot of people were thinking that, but they didn't want to say it for some reason. Can you talk a little bit about that? And like, how many other like archaic like lineages, that's what I'm gonna say, are we gonna find, if any?
I think that archaic lineages are going to start looking more like modern lineages, right? We talk about modern humans. We can talk about populations of modern humans, and we give populations names. And sometimes the populations give themselves names, and we like to use those as geneticists. We like to use those names. If people gave themselves a name, we'd like to use that one. We'd like to call the French people French, that sort of thing. But we don't always have that option. And sometimes when we're looking at ancient genomes that are ancestors of, to some degree, many living groups, we have to think through, okay, what do we call these folks, right? Are they associated with an archaeological tradition? That was the case with the Yamnaya, who became, sort of an archaeological population that got named for genetic reasons, and we can talk about as a group. I think that archaic people are going to start looking a lot more like this. Neanderthals. This is sort of like a racial name, right? We're talking about a giant group of people that at any one time probably numbered in the hundreds of thousands and were dispersed across half of Eurasia and had differences. If we looked at we know genetically, we look at the later Neanderthals, the Neanderthals that lived after, let's say, 60,000 years ago, you find that, they have some deep branches that look like they're 100,000, years, 120,000 years, sometimes older. That looks a lot like today's populations of Eurasia, right? If you look at people in Great Britain and people in Italy and people in Beijing and right all over Eurasia, you're looking at branches that go back 30,000, 40,000, 50,000 years, sharing an intertwined history, right? We understand that history is complicated. And I think that we're finding that the same is true of Neanderthals. With Denisovans, we know that our depth is a little deeper than we've found so far for Neanderthals, because the branch that mixed, at least two branches mixed with people today in Papua, in Australia, and in some other parts of Oceania, those branches look like they had a divergence from the Denisova cave branch that might be 250, 300,000 years old. And so that's as different as the most different living people are from each other. But we don't call living people different species. We understand we all interbreed with each other and we're part of a species. And I think that's also true of Neanderthals, Denisovans, right? I think humans are more expansive. And the problem of naming things. Okay, so you asked this great question about species and naming things, right? And from my point of view, I think that what we're going to find with these ancient human groups, like the Neanderthals, the Denisovans, is that we're going to start actually having more names for them in the same way that we have lots of names for living people, right? So we're totally comfortable talking about populations of living people with names. We like it when people name themselves. We like to call French people French, right, for instance. But the names that we use in the past, we often are talking about, the people that are associated with an archaeological culture. So something like the Yamnaya has a name, and that name comes from an archaeological culture. And sometimes we talk in terms of regions, right? So you have things like, Southeast Spain, sort of, Middle Bronze Age as a name, right? Names we use to try to keep track of what the entities are that are potentially mixing and adapting their environments and are connected with culture in some way. We have that with Neanderthals. We have different groups of Neanderthals. They were more different from each other across the range that they occupied, which is sort of the western half of Eurasia, they were a little more different from each other at any given time than most people are in that same region now, right? So their depth of diversity is a little higher, but not tremendously so. And they're associated with different cultures in that area. I have every reason to think that we're going to start actually differentiating these. There's no sense in calling them all one thing all the time, when in fact, it's different groups of them that factor into our ancestry and our connection with them. With Denisovans, the depth of divergence is even greater. It looks like 250 to 300,000 years for different groups that we've identified in genetic terms from their admixture with recent people. And so I think that we're probably looking at fossil groups that have some depth of diversity to them, that we'll probably recognize the anatomical differences between them. And as we learn more about them, we're going to have different names for them. I'm totally comfortable with the idea of dragon people as a name for these folks. And I love that in Chinese, this is Longren, right? I feel like Longren is a great name. And probably the Chinese Denisovans, we ought to just call them Longren. And maybe the Southeast Asian ones, we may discover that they're associated with the Nangdong sample. Maybe we'll call them Soloens or something, right? We're going to have names for things because as we learn more, we have to identify them. and that doesn't mean that the names have to be species names. I think the thing that most of my fossil friends get wrapped up in is having the right name for things. And the great privilege of working with genetics is that you can come up with a new name when the old one seems wrong. In taxonomy, the taxonomy of animals is tough to rewrite. Once you name something, that name sticks around. And we end up with all kinds of dumb names that way. We have names like Homo rhodesiensis, which is sort of an awful name, right? Rhodesia doesn't exist anymore. When it did, it was a highly racist state. We don't really want to talk about an ancient human group as having that as what we know about them. I think it's better to just use names that we can, that are flexible, that give us the opportunity to understand how they connect with each other.
Yeah, I think that's key. In terms of one of the things that I've taken away from your writings and your just talking to you and the thoughts is how people connect and how the lineages connect. I think one of the changes in the last 20 years is now that we know that most of us, actually all of us, probably have some genealogy from Neanderthals and they have genealogy from us. It's a lot more complicated and connected and unified, even though there are differences, obviously, cultural differences, biological differences. But this whole model of out of Africa 25 years ago, where it was like the super species that blitzkrieged everything we're not there anymore, right?
Yeah. I mean, the minute that you start actually looking at the details of this, right, you say, well, Neanderthals, they were pretty different from us in some ways. But if you inherited the OAS1 gene from them, you might have some ability to fight off viruses that most other people don't have. And so that's a pretty good thing. If you feel like, I forget which direction it is, right? But it's the circadian rhythm thing that the Neanderthal variation makes a difference today. And I don't remember if the Neanderthals are the morning people or the evening people, right? But the thing is that once you start connecting those traits, you begin to recognize, oh, okay, these traits are not me. I'm not the sum of these traits. And none of us are. And in fact, our ability to look at biology is very limited still, but we're learning a lot more about it. And the fact that you can look at UK Biobank and you can look at people's head shapes with MRI, and you can say, you know what? The Neanderthal heritage that they have seems to make a little bit of difference to how elongated or globular their head is. So it's there. That tells us about the connection. And I always think that breaking things down and looking at, how the parts actually work is much more helpful than looking at it from the outside and saying, I can't imagine this. I can't imagine how this would work, right? And that's where people were in the 90s. They were like, I can't imagine how the Neanderthals could be gone and we're here unless there was something really, really bad about Neanderthals. And today, the amazing thing is that people existed from African heritage in Eurasia for at least from 200,000 years to 160,000 years ago up until 50,000 years ago. Modern humans, right, African descendants, right, lived in Southeast and Southwestern Asia, in Israel, in as far to the west as Greece, and probably through much of Arabia and probably into Iraq and Iran, right? They're living in this area, and Neanderthals are there at their border the whole time, right? If there was a modern human advantage that caused people to just roll over archaic humans, they had 100,000 years to do it, and they never did. So, in fact, these groups were really, really similar to each other in their fitness where they were in contact. And those similarities, led them to have contact zones over time. We can see that they shared DNA sometimes. We know it made a difference to the Neanderthals. We know that in the end, when the modern group did begin to expand and expanded very rapidly, they brought on board around 2% Neanderthal and took it with them everywhere. So that's the dynamic. And those dynamics don't work when groups, species, whatever, are so different that they're incommensurable with each other. In fact, this is a biological situation where they were in contact and they were pretty comparable. And that comparability was what kept them in contact for so long.
Well, there's so many topics we didn't touch on, but I know you have to go. So I will let you go. We will do a follow-up conversation at some point, inevitably. It is as inevitable as admixture between different human lineages.
I really want to encourage people, hey, check out my Substack. I'm on Substack now.
Yes.
I've blogged for a long time, but Substack has been working really well, I have to say. I'm not shilling for them or anything. I'm just saying that, wow, people really find these. So that's great. And really hope that people will check it out.
Yeah, for sure. The links will be there, the show notes and everything like that. John's been producing great content for almost as long as me. Actually longer if you're talking about academic stuff, but those who know, and those who don't know, you guys should check out his work. There's just so much great stuff there, and you'll learn a lot. I've learned a lot through these podcasts, obviously, but also just like writing, like passively reading what you've written, John. And so I will let you go off of baseball practice. You have a good day, and I will see you online.
This podcast for kids